So Dr. Nicolas Casadei is a fellow Frenchman, just like me. So if there's another French person, tell it and we'll switch everything to French. Okay, maybe not, maybe not. But Dr. Nicolas Casadei did his PhD in Tübingen. He worked on an animal model for Parkinson's disease, and he developed behavioral tests and automation of measurement. He's now a research group leader on Parkinson's disease, at IMGAC, which is the Institute of Medical Genetics and Applied Genomics. And he's also the executive manager of the Genomics core facility at IMGAC, which means that we have the pleasure of working side by side with him on many projects. And we thank him for his advice every time. Nicolas is going to give an overview of methods used in molecular oncology. So on that note, over to you, Nicolas.
Thank you so much for the introduction. So it's my pleasure here to discuss a bit about genomics and oncology. As it was said previously, I'm like more a neuroscientist, and I already thanks Stefan Ozowski and Christopher Schroeder, who give me quite a lot of material and advice on this presentation. So briefly, the Genomics core facility, it's called CATG, and it's located in the Institute of Medical Genetics and Applied Genomics. It's located in the Frauenklinik in Tübingen, and it belongs to the University Clinic. And as it was previously mentioned, we, together with the microbiology of Tübingen and together with the Cubic, are part of like the NGS Competence Center from Tübingen. This is a network financed by the DFG. So our aim is mainly to promote genomic research on the campus of Tübingen, but also in Germany, develop methods that we can apply at the clinic. And our particular focus is implement transcriptomics in genomic analysis. The focus of the core facility concerning our service on campus and also with the DFG grant is mainly on the optimization of sequencing workflow. So we propose different tools and many options for project management, mainly from small projects. Larger projects are usually organized via Cubic. We have quite some specialty on nuclein isolation, also on library preparation and on sequencing. And at the core facility, we also propose some service on bioinformatics and data sharing, but which are rather based on collaboration. The Cubic is most of the time performing bioinformatics and data sharing when this is not performed on collaborative systems. A final slide about the core facility and to put a bit the context of this presentation. So the core facility is a mid-sized core facility where we process roughly 4,000 samples per year. We mainly work on human samples, but we still have a couple of model organisms such as mouse, bacteria, plants, a lot of rat work. And the diagram on the right, I'm not sure if we can see my mouse. Can I get some feedback if you see my mouse? No feedback. Okay. So on the right, basically, you see like the kind of service that we perform, and we mainly perform RNA-seq services. And one of the topic which is exploding right now, I guess, in all the core facilities, there is an increasing request of single cell sequencing, and we also have like an increasing request on long read and genome sequencing. So with that said, I would like to present you the genomic application, which were like the main reason around like this talk. So in the core facility, we propose methods which are separated in 3 different categories: a routine method, a research method, and a development method. Here I will start with our routine methods. So they are mainly derived from diagnostic protocols, but they are also applied heavily for many different research purposes. Typically, genome and exome sequencing, they are used a lot, particularly on rare disease. Here, one of the ideas is applying DNA sequencing in order to figure out specific variants. which are either observed in families or who could also be observed de novo. And here, something I would like to mention is this kind of strategy developed quite heavily in the last years due to the simplification of the data analysis. And I would like to show briefly what does it really means in the real research world. Here on this nice review is shown what happened when we do like a familial DNA analysis. So in a standard analysis, we would have different cofactors which would play a role on a specific phenotypes. We would have, for example, the population structure confoundings, which would play a role. We would have also assortative matings, which would play a role. Of course, parental would also play a role. And at the end, we would have like the direct effect from specific variants. But by performing trio-based analysis, we can normalize and remove almost completely everything which is based to the parental effect, but also to the population. And this simplified a lot the identification of variant for rare disease. Briefly, how work whole genome sequencing and also exome sequencing. So we start the work with a genomic DNA. This genomic DNA could be isolated from blood, for example, but could also be isolated, for example, from tumors. The first step we do is we cut DNA on small pieces. So here, for example, with an enzyme, so segmentation of the DNA on small pieces. And we would also do what we call the library preparation, meaning we will functionalize the DNA in order to make it amenable to sequencing. And here, what could be done, we could enrich for only coding region of the genome, for example. That could be done, for example, using a targeted capture, using magnetic bits. And that would allow to reduce the amount of data that need to be generated in order to find relevant variant. And then, of course, this is sequenced. Here, something which is important is sequencing is only the path for finding the causal variant. When sequencing a whole genome, several million of assemblies are usually found. Using Exome, many of these structural variant or like the SNPs are like not found. And we reduce the complexity of the data set by doing so. When we are focusing on all the variants which might play a role on the protein functions, we reduce significantly the amount of variant, but at the end, what we want is to find the fews which really lead to a phenotypes. And again, I would like to emphasize that using develop a strategy such as trio sequencing or different kind of cohort controls would help a lot to point out the causal variants. Also, something I would like to bring from our experience is the advantage of the whole genome sequencing when we compare it to the whole exome sequencing. There is a lot of work which is done on the development of a whole exome sequencing. These slides are a bit under bracket outdated from 2016, but we used to see a bit of a bias normally on the exome capture. And it makes some region of the genome a bit more difficult to analyze. When we see below the plot, we see that the effect is also not always very strong, but there are some specific application and some specific group of genes which particularly benefit of going to whole genome sequencing compared to whole exome sequencing. Also, the whole genome sequencing are a very good method for calling copy number variant. In this slide, what I show is the data we generated on prenatal data. So on red are all the structural variant, copy number variant that we missed using RS. And on green are all the ones that we could capture on RA. So what we see here is that the whole genome sequencing for capturing rather small copy number variants, so below 20 kb, perform much better than the RA. Another important point there is the importance of the depth of sequencing, particularly in oncology. There are classical tests which are done in human genetics, for example, in the context of rare disease, which are not very appropriated for oncology. In a standard basic analysis, where we would do 30x whole genome sequencing or 50x exome sequencing, if we would sequence a tumor, we might only pick, in this example, 2 different clones of cells. And these 2 different clones of cells during the progression of a cancer could be eradicated by a treatment. But during a relapse, for example, the focus would only be put on these 3 and only 1 could, for example, be detected. But in the optimal case, we would sequence much deeper. So that would be a coverage of several hundredfold on all genome and all exome. And there we could detect many more smaller cluster of a cell tumors. So here, by picking more clones of different tumors, we could also, for example, make better monitoring of the response of the tumor to treatments. And there we could also get more information in case of relapse to which kind of clones and which kind of resistance could have occurred. We perform normally in standard whole genome sequencing and exome sequencing, but we also have an onco panel with roughly 700 genes, where we target a depth which is between 500 to even 2000x, depending of the tumor contents and depending of the specific application. As I mentioned, the core facility has a strong focus on transcriptomics. And one of the reasons why transcriptomics might also be interesting on oncology is mainly on determining the deviation from the reference genome and also annotating specific variants. Here, what is particularly important is on whole genome sequencing and whole exome sequencing. Some structural variants are very complicated to pick by algorithm. I'm going to show examples later. And RNA sequencing can really help there. Moreover, there are also millions of changes which occur in whole genome sequencing, and several hundred of thousands which also happen on the exome sequencing, and some of them happen in regulatory region where it's a bit difficult to predict what would be the consequence of a mutation on like the expression of specific gene. So here the main application we used to perform RNA sequencing for investigating gene expression in the context, for example, of a pathway analysis of function of specific environments to the gene expression. Investigation of transcript isoform is also commonly done on RNA-seq. Particularly in oncology, the investigation of allelic expression is very interesting. In the case of somatic mutation, one could also explore whether the specific polymorphism or the specific somatic variation is really expressed at the RNA levels. Doesn't still mean that it's expressed at the protein level. And also we can investigate fusion gene, or at least validate whether fusion gene are really expressed. So there are many flavors of RNA sequencing. I don't really want to overwhelm you with that, but most of the time we start from tissue where we isolate RNA. On the RNA, we would digest DNA, and from this isolated RNA with DNA digested, we would have several strategies on how to continue the library preparation. The more brutal word would be to just sequence all the RNA you get. However, there would be what we call the broad transcription representation, meaning many different kind of transcript will be detected, and ribosomal RNA, which usually are roughly 80% of the RNA that we could find on a cell, they would be over sequenced and might basically make a lot of noise into the analysis. Based on that, we have strategy to reduce ribosomal RNAs. There, the advantage would be that we still have a large picture of what's happening at the cellular level, also with non-coding RNA, also including small RNA. But the main inconvenience of this strategy is always the contamination of DNA. So DNA is still detected in this kind of library preparation. And another problem that we have in this kind of library preparation is unprocessed RNA, like pre-spliced RNA. They are also usually highly abundant. Another strategy we propose to our user is also poly-A enrichment. By using poly-A enrichment, we decrease the amount of DNA into the sample. We also almost don't detect any ribosomal RNA anymore. And there, the only issue that we would have is if some transcripts are very highly expressed, they would be proportionally sequenced, meaning in the case, for example, of blood, we would see a lot of globin, which are available in this kind of total RNA. There is a hijack of this protocol, which is very convenient, where we only sequence the 3' of the RNA that allow to make relatively cheap gene expression analysis. And finally, the last method would be to capture cDNA of interest. But here, of course, the main inconvenience is one will need to know what exact RNA one is interested in. On the second part, I would like to show a couple of research protocols that we are also performing in the core facility and that could be applied in oncology. These are usually protocols which are developed during research projects. They were already tested and validated. However, because they don't run in large scale, usually they are still relatively flexible. A typical example of such experiment is long-read sequencing for DNA. There, usually the main aim of this method is to resolve complex rearrangement. The idea is when we perform exome or genome sequencing, in the normal case, we would, as I said before, get on the reference genome a piece of DNA which aligned to it. We would sequence a piece of the DNA in one direction, a piece of the DNA on the other direction, and by calculating bioinformatically the distance between the sequencing read, we would be able to map the reads and also give it sequence. But it happened that on the genome, some inversion might happen that could be detected by the fact that the two reads, instead of putting to each other, they would be misaligned. That would indicate to the person making the analysis that in the middle should be a breakpoint, and somehow this specific genomic region is reorganized with an inversion. Similarly, translocation could be seen by mapping on one sequencing read two pieces of DNA which don't belong to the same chromosomes. Or deletion could be seen by, during the mapping, observing that two fragments that should not be on the same sequencing reads are still together. So for solving these kind of problems, one of the idea would be to make what we called looking for split reads. That would mean trying to find the exact reads where the breakpoint occur. Unfortunately, these split reads with breakpoints, they usually need a very high sequencing depth, and they are, to our experience, not frequently seen. So in the ideal scenario, what we would like to have are long read of DNA that we can assemble together and to find this exact breakpoint and the exact genomic context. We currently use long-read sequencing such as nanopore for achieving such kind of performances. And here is, for example, a genome that we published from an A sample, so a reference genomes, where we could see that there are many breakpoints which could be we sequence we sequence a reference DNA, and we also sequence a tumor sample, and we could see that on the tumor cell culture we can detect many breakpoints with high confidentiality using long-read sequencing. Currently, in this specific presentation, I don't really want to mention too many companies with who we are collaborating and developing protocols. But in the case of long-read sequencing, it's a bit complicated to bypass that. Currently, are two main technologies which are used. The one that I already mentioned is a nanopore, where a long piece of DNA is functionalized in a way that it can bind to the bilayer of the nanopore, and the DNA could be inserted into a pore. When the DNA passes through the pore, it changes the potential of the pore due to some resistance of the electrons on these pores. And this reflect to change of current on the pores, which are detected by a computer and which are processed as a base calling. This allows to have very long piece of DNA which pass through the pore. However, the piece of DNA are only measured once. The speed through which the DNA pass through the pore is not really well controlled, and therefore it leads to quite a lot of error during measuring. In the other hand, another technology is to take is from Pacific Bioscience. The idea would be to take a double-stranded DNA pieces, ligate it in order to make circular a double-stranded DNA, and then having a polymerase which reads through it multiple times. That would lead to the strain which is forward and reverse to be sequenced. They could be aligned to each other, and all the small mistakes, which are called here subread errors, they could be corrected by bioinformatics, leading to also relatively long fragment of DNA, which could be sequenced in one read with relatively high quality. Both technology are currently available in the core facility and are running for human sample. We don't only sequence DNA with long-read sequencing. Another application we have is sequencing of RNA. Here, to make it short, we take a full-length RNA on which we are annealing on it a poly T and make the reverse transcription. We amplify this cDNA and we ligate on it the adapter, for example, here for nanopore sequencing. So this RNA transcribed in cDNA and amplified could then be sequenced on nanopore. And the advantage of it is, for example, here in neuroscience, HTRA2, which have different isoform based on different start sites. And here we can see that even with a not very high depth, 5,000 reads, we can clearly see on this specific sample the two different starting sites, one at the border with LOX L3 and one which is the annotated starting sites. These kind of events are quite difficult to pick up on short reads. And here we're indicating of different isoform of HTRA2 expressed in different tissues. Other application we also perform in research is the cell-free DNA. So cell-free DNA is the observation in the plasma of different DNA, which could come from different physiological events. Here, one of the most interesting one would be in the case of, for example, tumors with cell death due to apoptosis or necrosis, but also due to specific secretion mechanism, the detection in the plasma of these events. Currently, our main focus is to detect specific fragments of DNA which are coming from tumor which were already characterized and which could be used for finding these variants, into the blood. So the idea would be that for a characterized tumors which contain different mutations which were already observed after, for example, 2 lines of treatments, we would expect that the abundance of this specific fragment is extremely reduced in the blood due to the tumor, which is due to the tumor size, which is either gone or which is very small. And by monitoring the minimal residual disease, one could observe a potential relapse by monitoring this kind of variant. Here we develop a different kind of strategy in order to monitor this with high confidentiality. Stefan Owzowski developed multiple applications using what we call unique molecular identifier, which are added to the DNA, which is isolated from the plasma and which allow to correct with high confidence whether a specific mutation is an artifact or is could be really measured. These kind of projects are still for us research because due to the UMI strategy and due to the characterization of the primary tumors, one would run such kind of analysis currently as research project with start point and final point. Another application I mentioned before, which is also currently being widely used, is the use of single-cell sequencing, also in the case of oncology. Here, a paper we made again on neurosciences, where we were testing on cell extracted from embryonic mice, the development of the retina. We could identify a mechanism on which we could understand what are the progenitor of the retina and what are the different mechanisms involved into the differentiation of the retina. That could be also used in the case of oncology, for example, for resistance of tumors to treatment. And while looking at the resistant cells, trying to find the different cluster of cells which are resistant to a specific treatment and figure out which mechanisms are involved into it. Very briefly, some development we are currently doing. So these are pipelines that we cannot propose in streamlined service, which are collaboration with research groups which are either locally or which are nationally involved in our projects. We perform currently amplicon sequencing on the long read. To make it short, it's very helpful to resolve repetitive regions. And it's also very handy to find mutation on possible pseudogenes that could be performed with or without UMI, depending on the need of quantification. Epigenetic is also something we only propose right now in development, despite that there is, particularly in oncogenetics, more and more requests in that direction. Usually in epigenetics, the three protocols which are the most run are the attack sequencing, chip sequencing, and methylation analysis. In the case of methylation sequencing, there are three tests which are usually done. So there are methyl arrays, which are well characterized and easy to scale up. We unfortunately don't propose them anymore. What we propose are panels for methylation sequencing, on which we could focus on all annotated CpG which are known in human and in mouse. We also propose method for all genome bisulfite sequencing to find also new CpG or to find new methylated regions. The whole genome bisulfite sequencing is still a protocol which is in development. Chip sequencing is methods to figure out which histone marks might be dysregulated in a specific environment or due to a specific treatment. These kind of protocols, they cannot be afforded in research because usually the cross-linking of the DNA as well as the precipitation of the specific histone mark is something that has to be established. And we don't have enough experience on all the different kind of tissue and all different kind of antibodies. However, from the moment that the DNA is cross-linked and from the moment that the chip is performed, we could also help the scientists to perform the sequencing. And finally, the attack sequencing, which is something a bit alternative to the chip sequencing. On attack sequencing, the idea would be to figure out what are the boundary of the genome which are opened. And here, a bit similarly, the way the DNA is fixed and the length that the DNA has to be fragmented has to be optimized in every single experiment. What could see is that DNA which get under-fragmented or over-fragmented also leads to different profiles, and therefore this kind of bias need to be optimized. So would like to thank you for your attention on this project, and if there would be any question, I would be happy to answer them.
Perfect. Thank you really very much for this talk. I think it's so impressive every time I hear the different sequencing technique. I'm always amazed how fast it goes and how fast, like how far we've come in 40 years from the first sequencing to now all these different types and how we can really go into the nitty gritty of DNA. It's always really interesting to read about or hear about, of course. So if anyone wants to ask their question themselves, you can once again raise your hand or otherwise just ask your question in the chat and I'll be happy to read it aloud for Nicolas.
If I can just ask a question?
Yeah.
Nikolai, I was wondering, so you guys start to use more long-read sequencing, as you said. I was wondering if you, and also methylation analysis is interesting for you. So I was wondering if you actually use nanopore also for, or plan to use nanopore for also methylation analysis, because you kind of can do both with it. Have you tried that?
Yeah, that's true. So there are 2 points with the methylation and the nanopore sequencing. The first one is, of course, you need a larger quantity of DNA, because as soon as you amplify your DNA, you lose the methyl mark. So you need quite a large amount of DNA to start this protocol. But that said, once it's done, there are different algorithms which are there for calling methylation. And our first experiment with it is going de novo. It's a bit difficult. The statistics are not that good. Maybe if you go for like hypomethylated or hypermethylated, you might have a bit more success. But looking at fine dysregulation and also identifying exactly from which kind of profile the methylation signal is coming is also not that handy. The problem is the methylation does not change that much the occupiance of the pore, and therefore the difference of signal between the methylated and unmethylated base is also not that extreme. And finally, one thing also I have to mention, nanopore sequencing is extremely resource intensive. And currently we tend to avoid to do this bus calling because it just takes hundreds of hours to let this bus calling running.
Thanks.
And then we have a question from Gisela. She's thanking you for the really interesting talk. And then I wonder, how far have the long read and single cell technologies been applied for routine diagnostics, and what are the challenges there?
Well, I guess I don't take really a risk if I say that it's not implemented in routine diagnostic, and I don't think it's done. The point on the long read sequencing is still the turnaround time which is needed, and like the computing time which is needed. I think in the context of oncology, with phenomena which are a bit better known, it looks a bit more realistic. But when I'm speaking about a reassembly of a human genome, that usually take 1,000 hours of computing time. So oncology needs usually very fast feedback in order to adapt immediately the treatment. I don't think that that would be right now with the tools we have the most promising ways to go. Concerning single cell, there are actually application in the direction of immunology, which are getting more and more mainstream. But I think there, like the two issue are like the cost of the experiment, which are still extremely high, like 3,000 to 5,000 euro per sample, depending of the protocol and the depths needed. And there are still huge issues with the exact logistic of single cell, meaning the quality of the cells, the number of cells, some possible contaminant, and so on. So that's the way we would like to go, and that's the way we would like to develop. But in short term, I see these topics a bit critical.
Okay, we have two last questions. One is from Madalena. She's asking, what tools, software do you use for the analysis of alternative transcripts?
For the alternative transcript, so right now we use after the map calling tool, which is called the subread. However, I think our specific pipeline running Mexap is interesting tools that we run internally, but I think Cubic, which is running the CF core, they might have other tools which are maybe a bit simpler to run or maybe easier to implement.
And then one last question from Michael. Is there a sequencing method to detect RNA modifications?
Oh yeah, definitely they are. Like the one which is discussed the most is of course the nanopore, as we just said. So I think like the direct sequencing of single molecule in a pore, it's something extremely exciting, and it's something that we are definitely eager to collaborate on. Stefan Ozowski, who is in our institute, the head of the bioinformatics, has a lot of experience, not only on human, but also on bacteria and different organisms. But what is done mainly is precipitation. So what people do is they just use antibody, which pick specific RNA modification. They do an enrichment of their RNA that way, and then they go downstream with the preparation, something a bit similar, if you like, to chip sequencing where you have antibodies which allow you to detect specific nuclein modifications.
Okay, perfect. On that note, thank you again for this really interesting talk, Nicolas.